we're going to move on to Ryan Harvey. So Ryan's from University of New Mexico, and we'll be talking about hippocampal replay alterations following moderate prenatal alcohol exposure. Go ahead, Ryan.
Okay, yeah, thank you for the introduction. Yeah, so today for this, I'll be talking about hippocampal replay and sharp wave ripple activity following moderate prenatal alcohol exposure. So kind of as a broad background, globally around 9.8% of individuals drink or consume some amount of alcohol during pregnancy. And so up above, I'm just showing kind of the world map color coded by percent of individuals that drink per country. And this is an issue because even in pretty moderate amounts, and when I say moderate, I mean kind of like around 2 standard drinks in a day, so well below like the legal limit, you do see effects in hippocampus and other areas. So for one, there's a recent study that came out that showed even given a moderate amount up into adulthood, you see decreased numbers of GABAergic interneurons throughout the entire hippocampus. Also, there's been several papers showing reduced dentate gyrus LTP, and there's been several studies looking at how memory performance and spatial memory in particular is at a deficit following moderate amounts of alcohol. And so a recent paper that came out of our lab is we recorded hippocampal place cells in a rat with moderate amount of alcohol exposure. And for the rest of the talk, I'll just call it PAE for prenatal alcohol exposure. And so recording the place cells, we found that their place fields were slightly unstable over time in a couple different environments. Right here, we have a cylinder, and here we have a linear track. And so the field was kind of moving around slightly over the duration of the session. Also, along with that, we recorded or we measured the intrinsic theta frequency of the individual units and found that following alcohol exposure, the intrinsic theta rhythms were actually slowed down or decrease. And so when you consider theta sequences, that has some pretty broad implications to have a slower theta rhythm. And so with that kind of background, leads me to my broad aim is to explore the integrity of sharp wave ripples and replay events following prenatal alcohol exposure. And so gone over pretty well in the previous 2 talks, sharp wave ripple events and replay have been really implicated in memory. If you lengthen the duration of the ripples, you can recruit more neurons, pyramidal cells, and interneurons, and actually might increase memory performance. Alongside that, if you kind of truncate or shorten the duration of ripples, you can decrease memory performance. And because with this PAE, you do see decreased interneurons, as well as you do see decreased memory performance, I expect that these ripple events and this replay events will be somehow altered following alcohol exposure. So going to my methodology, throughout gestation, we exposed the rats to around 5% of ethanol, which is a pretty moderate amount. They're born, aged to adulthood, and we implant their hippocampi with electrodes to record single units and LFPs. And the task is fairly simple. First, we put them on a pedestal where they kind of hang out for a little bit of time while we're recording them. We drop them into a linear track where they kind of just move back and forth and have some time at either end to eat some Fruit Loops, and so we can record some sharp wave ripples as they occur. And after this, we put them back on a pedestal. So kind of like a traditional kind of paradigm for this. So getting to the results of this, what you see here are 2 ripples, one from a control rat and one from a PAE rat. And the white is just the wideband signal over the spectrogram, and beneath that is the bandpass filter signal. And from these ripple events, you can kind of take out several different features, such as duration, or like how wide they are, the frequency of the ripple, the amplitude of the ripple, and like the ripple interval, just to see how they're doing between groups. But along all these dimensions, I didn't find any differences between the 2 groups. So if there are any differences caused by PA, they're not along the dimensions of the ripples themselves. So the next thing I did was look to look at what the cells are actually doing during the ripple events. So on the left right here, we have an interneuron, and the right we have a pyramidal cell from hippocampus. And you can see that both these cells are actually pretty highly modulated by the sharp wave ripple events, and that they fire maximally during the ripple. But when we kind of move out and look at all the cells combined between or across groups, you can see that there is slight decrease of modulation of the fast spiking interneurons in the PAE group. And there is slight decrease in the pyramidal cells, but just not to the same extent as the interneurons. And alongside that decreased modulation, I did find fewer significant interneurons that were modulated by the ripple events. Okay, and you might have the next question of how does this affect hippocampal replay when you have decreased modulation of the interneurons and fewer interneurons being recruited into the replay events. And so here's like a kind of standard schematic where I have a good number of place cells, and here are their tuning curves. On each row, we have the cells raster over a good amount of time. And from both these things, we can use a Bayesian decoder to decode out their current position. And white right here is the actual position of the rat as they make their way down the linear track and kind of have a rest point right down here. And if you kind of squint your eyes and look in here, you can see there's about 3 kind of bursts of activities. And so those would coincide with sharp wave ripples. And if we zoom into this last one right here, you can see that the cells are kind of firing in this sequence, and using a more finely tuned or more temporally precise Bayesian decoder, we can decode out this trajectory that the rat is kind of possibly thinking about making as it goes back to the other side of the track. And so from those trajectories, we can, again, kind of pull out several different features, such as linear weighted correlation. And right here is just represented in absolute value, the distance of the trajectories, the speed, and the step size. And while the effect sizes between the groups are fairly small, denoted with Cliff's delta right here, I do see like the general finding of a truncation of these trajectory events. And so kind of meaning that while the controls kind of have, on average, more of a complete kind of representation of the entire track during the replay, the PAEs kind of have more stationary replay event. Okay, to summarize all those findings, first, the ripple features seem pretty similar between the 2 groups. Also, the interneurons are recruited in fewer numbers and with decreased strengths into the sharp wave ripples. And finally, the replay trajectories are slightly truncated. And so the next step I'm going to take is to go in and quantify interneuron and pyramidal monosynaptic connections to see how precisely these interneurons are kind of affecting these pyramidal cells during the sharp wave ripples, and maybe that might help explain why the replay events are slightly truncated. And so with that, here's my acknowledgements. I'd like to thank my team and collaborators and my funding source. Thank you.
Thank you, Ryan, for that great talk. We're just going to wait a second for a few questions. This is a small question from Kuder Bilal. Sorry if I butchered that. So when looking at your graph showing interneurons firing relative to the SW-Rs, control seems to be firing rhythmically, but not for PAE. Do you have any explanation for why that might be?
Yeah, so that was one of the big things that I found when I'm doing these analyses. I guess the traditional thought is during the replay events, the rats are kind of motionless, and so theta powers could be down. But for whatever reason, the fast spiking interneurons that I've classified, they were still theta modulated around the sharp wave ripple events. And when I made a null distribution, kind of like taking the same amount of ripple events, but just like taking random points in time and recreating those plots, it was a pretty standard kind of flat line. And so my control cells were modulated in the ripples, but also had a slight theta modulation, which I didn't see in the pyramidal cells. And I looked at the literature and didn't find too much about that, but maybe you have something to share with me, or someone does.
Yeah, I guess if anybody in the audience has any insights, we'd love to see it in the chat. In the meantime, maybe we can address this next question from Emily Airy Jones. Have you looked at slow gamma during the SWRs in the PAE rats?
Yeah, hi Emily. No slow gamma so far, but that'll definitely, that's on my list of something to do next. Yeah, but great question and great line of research to look into.
Yeah, that does sound like it'd be very interesting. Emily says hi back. All right, cool. So we're just running into the end of this session. I'm going to let people send in a few more questions if they'd like. Technically, we're ending now. It's 2:45, but we'll just wait for a second. In the meantime, I'd like to thank the panelists for their fantastic talks. And I really appreciate the engagement of the audience here too. So good job all around, guys.